Hi class, so I'm posting this to help you maybe to understand how I was expecting you to do your independent research on topic BLAST. So I hope this will be helpful for you in future as well. So let's start from the very beginning. The topic that you had for your independent research was BLAST. And me as a student, when I hear a topic, and most likely it's a known topic for me, the first thing that I would do, of course, I would go to Google and I would type BLAST. Of course, you were given some materials to read. Either way was okay to start from that material or just do your own like independent research from the very beginning, from scratch. So when you Google BLAST, this is what will come up. So basically, you can see immediately what is BLAST. BLAST is Basic Local Alignment Search Tool, right? So, and it is an NIH tool, and it is available for everyone for free to use. So here we go. We can open it on your browser, and then there are different options here. We can blast nucleotides, we can blast proteins, and by now you knew already that the gene sequence can be presented in nucleotide order sequence or in protein format where you have amino acid sequence. So either way is fine. The task that I have given you was in nuclear base order. So for that, of course, you are going to go to nucleotide base. And all you really had to do was to go to the file that I've sent to you, which was, it was opioid receptor kappa 1. So at this point, you didn't even really need to, let me see what happened to this. Enable. So you didn't even really need to know what is opioid receptor kappa, right? However, we will talk about it further as we move forward with this. So what do I do first? I just copy the first gene that I see here. Copy and paste it here. So here are different options. There is BLAST-N, which is for nucleotides. There is BLAST-P, which is for proteins when you have an amino acid order, and so forth. So really, those are the two that you will initially use the most if you are dealing with gene alignment and gene sequencing. So when you have copied your gene sequence here, all you have to do is to push BLAST. So because it's going to take some time to do the inquiry, I already have done it here. So this is the result that you are going to see here. Let me just close this one so it will not use the whole capacity of the computer. So this is the result that you are going to see. For the first sequence, this is because it was not only the part of the gene that I gave you to sequence, the opioid receptor, but also it had some other things on it. That's why you are seeing these parts from 1 to about 2000. And then after that, all these lines that you are seeing are more representative of your opioid receptor kappa gene. So here you go here and you are seeing the similarities that BLAST found, the program found with different genes. Of course, the first one that comes is the Homo sapiens, and then it has some similarities with other genes as well. The second one is a monkey gene, and so on and so forth. So this is what I was expecting you to do, to run the BLAST and just basically report the data that you have gotten here. So this is for the second one where we had the mouse gene. This is the results for mouse, and it recognized as a mouse, and it found some similarity with other genes as well to different extent. And it was up to you to report, as I mentioned before, there is not right or wrong answer. It's up to you how much of information you want to get from here, how much information that is out there you want to use. And then finally, the third one was the monkey gene. And the results for it was that it was 99% Homo sapiens human, and it was about 100% similar to monkey gene. So this would have been good enough information to receive. However, if you want to do more digging in, if you have more curiosity and you are like, okay, what is this opioid receptor kappa? I would absolutely encourage you to go ahead and search. The first search again you would do would be probably in Google, right? And you would just put opioid kappa, for example. You can get different articles about it. If you don't want to dig in that far, just searching opioid will bring you to probably opioid receptor. Here we go, Wikipedia. Definitely, I'm pretty sure Wikipedia has a lot of information on opioid receptors. And the first thing that you learn about it is that it's a GPCR. It's a G protein coupled receptor. It's a very large class of receptors. And I mean, this is a place where you can dig in and it can take you farther and farther. And it's always up to you how far, how deep you want to go into this. And then, like, of course, okay, the type of receptor this is, opioid receptor, it's a G protein coupled receptor. So it's a GPCR. That's a very specific type. And there are databases for gene sequences and tools to align and get the sequences of those genes already existing. So one of them is, for example, the GPCR database.org, where you can get all information about all different receptors. You go to the receptors, you can search structure-based alignments, for example, or just do a very simple receptor search, for example, here. And you can just put your receptor or just like find from browser here what type of receptor you are looking for and do the alignment. So here I want to show you how does in general, for example, working with GPCRs, how does it look. The sequence here is given is for mu opioid receptor. This is the amino acid sequence. This is the protein, how the protein looks. This is the structure from the top. So GPCRs are 7 transmembrane receptors. So they start from outside of the cell surface. They go into the cell surface, make a loop, come out, go in, make a loop, come out. And basically this is how they look. And in the middle is the membrane, basically this big ECL2, which is extracellular loop 2. It is basically located outside of the membrane, and then the same is for extracellular loop 1 and extracellular loop 3, and this is intracellular loop 1, 2, and 3. So when you have information about gene sequences and you are aligning them, you can see, for example, that let's say this tryptophan at the position, and this is fun tool, you can choose a color, for example, and you go and you're like, okay, tryptophan at position 295, and this is the protein structure, remember? So the positions are of amino acids. At this position is remaining the same for opioid receptor mu, kappa, delta. So we call it a conservative residue. for example. And I mean, and there are tons of information, and it really depended how far you want it to go and towards which direction you want it to go. So this is one website that you could have used for that purpose. Another website that, just like searching, you could come across would have been this European Bioinformatics Institute's website, which is also providing tool for gene alignments. And this is how your aligned genes would look here. Or another place, again, from that same institute would be the cluster omega, where you could put here you can choose, do you want a protein sequence? Do you want a DNA sequence? Do you want an RNA sequence? And you copy and paste your sequences here. Sequence in any supported format is okay. They are like very generous. You don't need to do a lot of reformatting to be able to use this tool. And then as a result, you are having something that looks like this. I'm not going to run it right now because it will take a bunch of time, but this is how basically your workflow should have been. And also, just maybe to satisfy your curiosity, I can show in real world how, when you run all of these sequence alignments, and then how you can look at them and what's the point of doing them. I know I talked about this during the first class, but again, I will show you some examples. For example, here is an alignment of human and dog arginine vasopressin receptor 1. And like here, you can see, for example, those are conservative residues here, N, V, T, K, along both genes. So query in the subject gene are remaining the same. They are remaining here, NVTKA11. They are remaining conservative. They are being the same. Or another example, actually just like final, how does it look, human and dog alignment. And knowing a lot of information from the snake structure. Here, this is what the snake structure is. This is the visually, you can see where the residues are sitting. And looking at that, you can think, definitely, if there is a drug that is interacting with this receptor and drug cannot penetrate through the membrane, definitely it cannot link to amino acid here or here, but it can definitely interact with amino acid here when the amino acids are still on the surface of the membrane, and the compound, the drug can reach those, right? So taking all of this under consideration, and also the physical chemical properties of the amino acids, which one will know doing the research on this topic, this is what I have done. I have went and I said, okay, so we are looking at rat, human, and I think this was dog, the third one. And we are looking at that and we are saying, okay, at this particular position, there is for rat, we have V, and for human and for dog, we have I. Maybe this is something that would be interesting to mutate and see if it changes the interaction. And the same is here. We have V, I, and we have L. So all three species have different residue at this particular position. Here, again, human and dog have the same E residue, and rat has different residue, or in this case here, AGA, where rat and dog have different residue from human, where human has G. So, and this is all very informative for research purposes, but this is something that, again, it's up to you and up to your work eventually, how far you want to dig into all of those. I hope this was helpful. If you have any questions that you think might be useful to address, please email me, and I will try to help you with figuring this out.